Today we are going to be looking at respiration and we're going to see how it goes because respiration is a huge topic and we'll get done what we can get done. There are more of these A2 revision sessions and there's a good chance that this may carry over into a future session as well. But let's see how we get on. It's such a big topic and it's such a key topic. It's a topic you know you're going to be tested on. What we're going to try and do today is focus on the real essentials, the reactions that are involved, and we'll get some opportunities to apply that, I hope, as well. Right, okay, so here's the plan. We're going to look at each of the stages of aerobic respiration, trying to understand them rather than just basically recall them. Recalling will always be more difficult. If we can understand them, that's going to work a lot better. We're going to look to be able to identify the different stages and the reactions of respiration in novel questions. So we'll look at some questions that will look a little bit different to what we've seen and try and match up what we've seen to those questions themselves. And we're going to finish, hopefully, depending on how we get on, to look at the yield of ATP from each of the stages of respiration. And all of those things really tie in together, really, because it's about understanding the different stages so we can apply them to something new and be able to deal with those questions that come up, which ask you to really make sure you understand the different parts. So, okay, let's do the essentials to begin with. So this is what we got from GCSE. Glucose plus oxygen gives carbon dioxide and water, and you might have in brackets there plus energy, or alternatively, you might have energy coming out as an arrow. You might have ATP instead of energy, and all that's fine. But the essence we know from right back from year 7, glucose plus oxygen gives carbon dioxide and water, and the whole point of that is to release energy from food. So this might be the sort of thing that you'd have in your notes or in a textbook. Let's have a look at these rather than me reading this out. Let's have a look at each of the things in detail to provide us with a foundation from which we're going to build. So to begin with there, we've got this idea that respiration is a series of enzyme catalyzed reactions. So rather than it being a single reaction, like is shown at GCSE, actually it's multiple reactions that are involved, each one being catalyzed by an enzyme. It says we're energy rich respiratory substrates. Well, we could say we're glucose, because glucose is the main substrate we're familiar with, but there are other substrates that can be used in respiration, but we will focus on glucose today. But it may be something we look at in the future as to what other substrates can be used as well. And then what happens to those substrates is they're broken down to release the energy. Now that energy, this is our goal, is to trap that energy as chemical energy in ATP. That's really what we're trying to do, get as much ATP as possible. ATP, that universal energy currency that's formed as a result of chemiosmosis. So sort of what you might cover before you do respiration, really, to be a chemiosmosis resulting in the formation of ATP. And the idea being, remember that ATP then is used, it's that energy currency because it's used in all cells, all reactions, all organisms, in order to carry out any energy requiring process. Active transport, muscle contraction, protein synthesis is needs ATP. But in doing so, some energy is released as heat. So it's not perfectly efficient. There's going to be some energy losses as heat too. And the next thing it says there, look, during respiration, high energy carbon-carbon, carbon-hydrogen, and carbon-OH bonds are broken. So here's a glucose molecule. Again, we're going to focus on glucose. You can see a carbon-carbon bond. There's plenty of those in there. We've got a carbon to an OH group up there, and carbon to a hydrogen bond as well. All those bonds contain energy, and glucose contains lots of energy because it has lots of those bonds. And what we do as a result of that, when we break those bonds, is that we produce other molecules, carbon dioxide and water, which have lower energy bonds. So if you add up all the energy that's found in all the bonds of glucose and oxygen, and you add up all the energy of all the bonds found in the carbon dioxide and the water that's produced, we'll find that the glucose and the oxygen mainly the glucose really, contains a lot more energy, or the left-hand side contains a lot more energy than what's on the right-hand side. And that difference in energy is what's used to make the ATP. That difference released is used to attach inorganic phosphate to adenosine diphosphate, ADP, to make ATP. So there's, if you like, the essentials, the background. But really, we need to go on a step here. So let's now look at words I'm sure you're all familiar with, but we'll deal with each of these in turn today. The four main stages of glucose breakdown. Okay, so glucose and oxygen being converted to carbon dioxide and water. So we have glycolysis, link reaction, Krebs cycle, and the electron transport chain. It says four main stages. Sometimes it's referred to as 3 stages because the link and the Krebs cycle are quite often grouped as a single stage. It doesn't really matter. I've got a picture of a cell on here because it's important that we appreciate where these things are taking place, something I'll refer to again. But glycolysis, that first stage, takes place in the cytoplasm, or the cytosol as it's written here. It takes place in the cytoplasm, whereas all the others, the link, the Krebs and the electron transport chain, take place in the mitochondria. And it's an interesting idea really that we're used to, and I talked to you, said at the start about trying to make sure you understand what's going on with these different stages and the reactions. That actually it's quite common that, no, not common, rather, it's what we would expect is for all of respiration to take place in mitochondria, because that's what we're used to. At AS, we would have said that. At GCSE, we might have said that. But actually, it turns out that the first stage doesn't take place in the mitochondria. It takes place in the cytoplasm. Link and Krebs take place in the matrix of the mitochondria, and the electron transport chain takes place on the inner membrane of the mitochondrion. Okay, so without further ado, let's begin with glycolysis. And as I just mentioned, sort of the odd thing about glycolysis, and we like to see odd things because they become memorable, that takes place in the cytoplasm. So we've kind of become used to this idea that saying that respiration takes place in mitochondria, but therefore the fact that our first stage taking place in the cytoplasm is a little bit strange, but that's fine. It makes it memorable, so it's not a bad thing. Let's begin with what we would expect to begin with is glucose. So glucose is our starting molecule, as you might expect. Now in our equation we'd say glucose plus oxygen, but there's no oxygen used here. We just start with glucose, which is a 6-carbon carbohydrate. We need to be tracking the carbons as we move through this series of reactions, because it helps us to fit in other intermediates, to fit in other molecules. So the first stage is that glucose is converted into this molecule called glucose phosphate. In fact, we say glucose is phosphorylated because a phosphate is added to it. In order for that to happen, we are going to use ATP. So I just spoke about understanding and recognizing when things are a little bit strange because it makes it memorable. This is an odd idea. But why is it that we're going to use ATP in respiration when the goal of respiration is to actually produce ATP? So it is strange that the first thing we'd do is to use up ATP, but there's a reason for it, which we'll come back to. So the ATP, you can see it's got 3 phosphates. It's going to be converted into ADP, 2 phosphates or diphosphate. And so where's that 3rd phosphate gone? It's been added onto this phosphate, to this glucose molecule. Glucose phosphate is also a 6-carbon molecule, which makes sense because we can't see any reason why we would say there's been an increase in number of carbons or a decrease in number of carbons. So we would expect it to be a 6-carbon molecule. It is just glucose with a phosphate attached. Next stage is glucose phosphate is converted to fructose bisphosphate. Now bisphosphate, you might expect it, or it could be referred to as diphosphate. Diphosphate makes a little bit more sense because we know that diphosphate refers to 2 phosphates, but actually it's referred to as more commonly as fructose bisphosphate. The fact that I'm saying there's 2 phosphates makes it very logical that we have the same thing happening again. An ATP molecule is converted to an ADP molecule, and that phosphate, that 3rd phosphate, is added to this glucose phosphate molecule. So now we have 2 phosphates here, the bisphosphate. The reason it's called bisphosphate or not diphosphate, not that it really matters a great deal. You can think of it as biphosphate if you like, but Bis means that those two phosphates are on a different part of the molecule. The two phosphates are on either side of the fructose molecule. But it's not called glucose bisphosphate, is it? It tells you this something else has gone on there. That glucose has been converted into fructose within that process. Fructose is an isomer of glucose. So as you know, we're going to see exactly the same number of carbons, hydrogens, and oxygens. It's just going to be rearranged. But it's still going to be 6 carbon because, again, we can't see anywhere where those carbons would have been added or taken away. That stage there, where we're using ATP, that odd situation, we're using ATP in a process which is intending to release or to produce ATP, is called the preparatory phase. So yes, there's an energy input to begin with, but we are going to liberate some ATP in the remaining part of glycolysis. So here we are at fructose bisphosphate. It's unstable, so it naturally breaks down to 2 molecules called glycerate 3-phosphate. Now, if I asked you to think about how many carbons there are going to be in those 2 molecules, you can just see, well, there were 6 carbons, and now it's been split in 2. It's the same molecule, so it must mean that those 2 molecules must have 3 carbons, and they do. The 3-carbon carbohydrates, they are referred to as a triose. So at AS, we talk about triose sugars, and we don't really, all we say is there's triose sugars that are involved in metabolism, and that's it. Metabolism really just refers to all the reactions that take place in cells. But now we do see triose actually appear. Here's a triose, it's glycerate 3-phosphate. Look at the wording there, the 3-phosphate. You could be forgiven for thinking that involves that means there's 3 phosphates on each, but that wouldn't make sense because we've only got 2 phosphates on the fructose bisphosphate. So 1 phosphate goes to one side and 1 phosphate goes to the other. So each glycerate 3-phosphate molecule has a single phosphate each. The 3 refers to the position on the on that molecule where the phosphate is found. I wouldn't say it's especially important to remember that. Those doing chemistry might be more familiar with the idea of numbering carbons, but for biology it's not necessary. But know the name, that's all, glycerate 3-phosphate. Okay, so now we're going to get some liberation of energy. So glycerate 3-phosphate, 3 carbon, is converted into a molecule called pyruvate, or pyruvic acid it could be referred to as well. Now in order, that will happen twice, happens on both sides. In order to make that conversion, a number of things are going to happen here. So first of all, we're going to get some liberation of, or we're going to get some production of ATP. So this makes perfect sense. I've got adenosine diphosphate here, and I've got a phosphate on the glycerate 3-phosphate molecule, and it's going to take that phosphate and form ATP. It happens again. So we're going to get 2 of these ATPs being formed. The second one makes less sense. Now, I'm a big believer that everything should make sense here. So you should be able to track through the number of carbons. You should be able to track where those phosphates are going. But there are occasions in respiration and in photosynthesis actually where things just don't quite fit. It's not a perfect match, and you can't follow through all those molecules. And this is one of them. What you could say, I mean, strictly speaking, the way it should go is rather than it just going to, we'll do the second one, ADP to ATP, it should be ADP plus PI on the second one, because the phosphate has to come from somewhere and it would come from within the cytoplasm. But it's never something you're going to be asked about. You might see ADP plus PI, you might just see ADP. You know what's going on. The important thing is we're releasing ATP at that point. The other thing that happens, something a really important stage now, is that in that conversion of glycerate 3-phosphate to pyruvate, a molecule called NAD is converted into reduced NAD. I've abbreviated it here as rNAD. So whenever you see rNAD, and we will see it a lot, you can refer to it as reduced NAD, or know that it is reduced NAD, and I will refer to it as reduced NAD. Now, what is NAD? So a common question, what is the role of NAD? It's a hydrogen or an electron acceptor. It's up to you which one you use. This is what's going on, and there's a lot of chemistry going on here, but really we've got NAD, which will, it can accept a H+ ion or a proton, and it can accept an electron and form this NADH or reduced NAD. Now, there's too much chemistry here for us to really need to go into the detail of this. So That's why I prefer to use the term NAD and reduced NAD, because we don't have to deal with this NADH as it's written there. Now, the NADH can be written, or reduced NAD can be written in various different ways. You could call it NADH. You might see it as NADH2. You might see it, I think the correct way is NADH plus. But all that is just a level of detail that can be confusing. So I will keep referring to it as reduced NAD. So what you need to know is that NAD is a hydrogen or electron acceptor. You might benefit from knowing that when NAD is converted into reduced NAD, it accepts a hydrogen, and that hydrogen is composed of a hydrogen, a positively charged hydrogen ion and an electron. But that might be useful later on. But the key thing is to know that NAD is converted to reduced NAD. So in order for that to happen, in order for NAD to be converted to reduced NAD, it's got to accept this hydrogen. And where's that hydrogen going to come from? Well, it will come from the glycerate 3-phosphate. As a result, we say this reaction is a dehydrogenation reaction, because hydrogen is being taken from the glycerate 3-phosphate. So whenever you see NAD be converted to reduced NAD, we will know that that reaction is a dehydrogenation reaction, and the glycerate 3-phosphate is being dehydrogenated. Common question, so worth sort of making sure we got to grips with that. Okay, it's a dehydrogenation. Pyruvate is 3 carbon, because we can't see any way where carbon has been taken away from this, so it's 3 carbon to 3 carbon, and that happens on both sides. And it's an important thing to remember that from now on, everything's sort of happening twice, because we've got stuff going on with glycerate 3-phosphate, and what happens from there down. Pyruvate will now go into the Krebs cycle, so that's going to happen. That pyruvate will go into the Krebs cycle, or Lincoln Krebs, and so will this pyruvate as well. The other thing worth mentioning here is this idea of what we call substrate level phosphorylation. So when ATP is formed, it can be formed in 2 pathways. It can be formed through substrate level phosphorylation or oxidative phosphorylation. Oxidative phosphorylation we'll come to later. Substrate level is where you see ATP being formed at any of the stages of glycolysis, the Lincoln reaction, or the Krebs cycle. If you're seeing ATP being formed there, it would be classed as substrate level phosphorylation. I would word it differently, I suppose. I'll say this: whenever we'll look at oxidative phosphorylation later, but oxidative phosphorylation involves ATP synthetase, which I'm sure you're familiar with. Whenever ATP is formed via ATP synthetase, it's oxidative phosphorylation. If you see it formed anywhere else, it's substrate level phosphorylation. So we're seeing ATP being formed. We've got 1, 2, 3, 4 ATPs being formed, but 2 ATPs are used up. So we've got a net gain here. Let's have a look at that. Our net gain from glycolysis is the 2 ATP molecules. I'll say that again. We've got 4 produced. 1, 2, 3, 4. Take away the 2 that have been used. So we got 2 ATP molecules that are produced as a result of glycolysis. We got 2 reduced NAD molecules, 1 and 2, and we got 2 pyruvate molecules. Now, here's an example of it's a past paper question, and look what you'll see. There's no question on here to do, but just notice the way it's worded. So this is, see, it says glucose here, fine. So it shows the outline of 3 stages of aerobic respiration. Okay, so they're showing glycolysis, link, and the Krebs cycle. But let's just focus on the glycolysis for now. We've got glucose, and look what they refer to here, triose phosphate. They haven't mentioned glucose phosphate. They haven't mentioned fructose bisphosphate. They haven't said there's 2 molecules of triose phosphate. They haven't called it glycerate 3-phosphate. So we've got to deal with that. You've got to deal with the fact that it won't be presented in the way that you might be used to. Here's another version where they go, look, here shows glycolysis, Lincoln Krebs, but let's look at glycolysis. They just call it a 6-carbon sugar, and now a phosphorylated 6-carbon sugar. So instead of glucose, they call it a 6-carbon sugar. You've got to do the work there and go, oh, they mean glucose. A phosphorylated 6-carbon sugar, oh, that's glucose phosphate. It might be referring to fructose bisphosphate, because the next one is a 3-carbon sugar phosphate. So you see, they're not using the wording that we would normally be used to, or that I've just gone through. So we've got to be ready for that. So for that reason, here's perhaps the other most likely way you'd expect to see it. So on the right hand side there, I've shown you, I've reproduced what I've shown you already, but look at the other way we could refer to this. So we could, rather than calling it glucose, we could call it a hexose, and we could call it then, instead of glucose phosphate, a hexose phosphate. That's fine. That's requiring you to know that glucose is a hexose sugar. Well, and indeed that fructose is a hexose sugar. Okay, it's 6 carbon, so we should know that. But see, it's working at a slightly different level. And now instead of glycerate 3-phosphate, they're referring to it as triose phosphate. Absolutely fine. We've got to be ready for that. So it's not a case of one being better than the other. We just got to appreciate that glycerate 3-phosphate and triose phosphate are the same thing. Or to be specific, glycerate 3-phosphate is a specific example of a triose phosphate. Triose is more of a category. It's a 3-carbon carbohydrate, whereas glycerate 3-phosphate is a specific 3-carbon carbohydrate which has been phosphorylated. So we need to keep that in mind. Right, so let's have a look at a question. So it says, diagram below is the outline of glycolysis. It's glycolysis alone. And look at it there. So we've got glucose, hexose biphosphate, and now triose phosphate and pyruvate. I'll give you a minute, have a look at these questions, and then we'll come back and I'll go over what you could have had. Okay, so the hard part of this is really, it's not the questions really, it's getting your head around which part of this pathway corresponds to the pathway you're familiar with. First bit's straightforward. Where the glycolysis takes place, no problem at all. Cytoplasm, no issues with there. Nice easy question. A little bit too much recall actually, so it makes it a little bit too easy. But now we've got letters A, B, and C from the diagram to where the following places take place. So dehydrogenation. So dehydrogenation takes place between the triose phosphate and the pyruvate. So that's the NAD collecting that hydrogen from the triose phosphate or the glycerate 3-phosphate. So we know dehydrogenation is going to happen at C. Substrate level phosphorylation, again, that formation of ATP occurs between triose phosphate and pyruvate. C again. Phosphorylation using ATP. So this is where ATP phosphorylates the glucose. You can kind of see it on there that glucose is being converted into well, in this case, a bisphosphate. So that's going to be A. And the splitting of hexose, where we've got a hexose down to a triose. So that should be B. If you don't know what you're doing, that's really tricky. If you can't make sense of the pathway and link that to what you know already and your version of the pathway, that's really tricky. If you can, it's very straightforward. State the net gain of ATP when one molecule of glucose is broken down to pyruvate in glycolysis. So we should know that that's 2 ATPs produced. Okay, let's move on. So we've just done glycolysis. We've just done stage 1. And I'm looking at the time we've got, and it's okay. I'm thinking we'll get done what we get done, which is why I said at the start, look, we've got 2 sessions. This may well run over 2 sessions, and it looks like it will. We're going to work on the link reaction, the Krebs cycle, and we'll see what we get done and see how much we can cover. So here we go. Now takes place in the mitochondria. Great. That's what we'd expect from respiration, but it's in the mitochondrial matrix. Notice that whenever it's not uncommon, and I'll show you some examples where you've got to specifically state where, I think the word is state precisely where a particular stage takes place. In this case, the precise location is the matrix of the mitochondria. We need both. Our starting molecule is pyruvate. So the pyruvate, which has come from glycolysis, it's fine. Remember, we have 2 pyruvates, but we'll just focus on 1. But we need to keep that in mind when we start looking at yield of ATP. So the first stage, pyruvate is converted to acetate. Now, the notable thing here is that acetate has 2 carbons. So we have lost a carbon between the pyruvate and the acetate, which means we know something is going to have gone on in between, which has caused that carbon to be lost. And indeed, we see the production of CO2. Well, this is quite good in a way because this links in with what we know already. We know the equation for respiration is glucose plus oxygen gives carbon dioxide and water. And until this point, all we've done is dealt with the glucose. Okay, we haven't dealt with any water and we haven't dealt with any carbon dioxide. So we're starting to see that now. Whenever we see the loss of carbon dioxide, we call it a decarboxylation. So we've had a dehydrogenation, removal of hydrogen. Now we have a decarboxylation, so removal of a carbon or removal of a carboxyl group. At the same time, we have NAD being reduced again to form reduced NAD. Again, this is a dehydrogenation. So this reaction is focused upon in questions because it gives you the opportunity to talk about the conversion of pyruvate to acetate, fine, but to use the term decarboxylation, to use the word dehydrogenation. In fact, the other thing that's often asked that now comes to my mind now is the enzymes involved doing this. So the enzyme involved in converting pyruvate to acetate, because it carries out decarboxylation, is referred to as a decarboxylase enzyme. And indeed, it's also referred to as a dehydrogenase enzyme. So it's an enzyme which has the ability to carry out both shows decarboxylase activity and dehydrogenase activity because it carries out decarboxylation and dehydrogenation respectively. So often questions ask some detail, 2 or 3 mark question on what goes on in this particular reaction. The next thing that happens is we have the introduction of this molecule called coenzyme A. Now, coenzyme A is not the first coenzyme we've come across. Actually, NAD is a coenzyme as well. Now, coenzyme is a molecule which needs to be present in order for an enzyme to carry out its reaction. But actually, I don't think I can even recall. I don't think it's ever been questioned as to what a coenzyme is. But to know that the role of coenzymes is actually widespread in respiration. We have coenzyme A, which is clearly a coenzyme because that's its name, coenzyme A. But we also have other coenzymes. We have NAD being the best example of that. So acetate combines with coenzyme A. It's a temporary association. It forms this molecule called acetyl coenzyme A. We still say it's got 2 carbons, even though coenzyme A actually might affect that. But because really it's such a temporary association with coenzyme A that really we're still focused on the acetyl or the acetate part. So it's 2 carbons. That stage there is called the link reaction. Okay, link in the glycolysis stage with the Krebs cycle. But quite commonly you will see, as I've shown here, link reaction and Krebs cycle together. Let's get into the Krebs cycle now. So first part of the Krebs cycle, if you can have a first part of a cycle, is the acetyl-CoA now combines with this molecule called oxaloacetate. So the name you do need to know. It's 4 carbon. Again, there's no reason you should know that. There's nothing instinctive in that. You have to know that oxaloacetate is 4 carbon. It combines with this 2 carbon acetate and it forms this molecule called citric acid, sometimes referred to as citrate. And you can guess the number of carbons on that because I've got 2 carbons from acetate or acetyl-CoA and 4 carbons from oxaloacetate. So combine those together, I get 6 carbons. I said the association with coenzyme A is temporary, and indeed, once those have been joined together, the coenzyme then is released and can go on and do its job again and again, combining with an acetate and facilitating this combination of the acetyl part and the oxaloacetate to form citric acid. So here we are. So this first major reaction, oxaloacetate to citric acid. The citric acid actually it seemed to be quite a key molecule. In fact, the Krebs cycle is sometimes referred to as the citric acid cycle. From there, from citric acid, it is converted into a 5 carbon compound. Now it has a name. Thankfully, we don't need to know the names of all these molecules. We do need to appreciate, though, simply that it's a 5 carbon compound. I don't need to add on that it's a 5 carbon because it's called a 5 carbon compound. That's what we will refer to it as. But clearly, there's a loss of a carbon. So what are we going to see? As you'd expect, CO2 is being released. It's also a decarboxylation reaction. What do we also see? A dehydrogenation reaction at the same time. So we've got NAD picking up hydrogen, dehydrogenating citric acid. So it's picking up hydrogen from the citric acid and it's forming reduced NAD. And now this is going to become common. Okay, we're going to see these things over and over. The other thing that happens here is we have an input. We have a input of water, and that's a little bit strange to see because we know that respiration releases water, and so it's quite odd to see water going in. What we do know is that ultimately we're going to get the release of water. So at some point this is going to have to be paid back, if you like. Next stage, we have a 5-carbon compound being converted to a 4-carbon compound. Again, we've got a loss of a carbon. That makes things easy. We know there's going to be CO2 lost. We also have NAD being reduced again. So this is now a common theme. We can get used to this. It's quite nice in a way that we're just seeing the same thing again and again. We also see the water going in. Brilliant. We've seen it before. So we know the first and the second stage at the moment look identical. The only thing that happens is we've got a different number of carbons in those intermediate molecules here, the 5-carbon and the 4-carbon. Here we do get something different though. We do actually get the formation of an ATP molecule through substrate level phosphorylation. I know it's substrate level phosphorylation because there's no ATP synthetase involved. We just have the combination of ADP plus an inorganic phosphate to form ATP. Next, the 4-carbon compound is converted to a different 4-carbon compound. Are we going to see the same thing again where we've got water coming in? We're not going to see the loss of CO2 now because we've got the same number of carbons. What we are going to see is another dehydrogenation. But in this case, we're going to see it differently. We're not involving NAD. We're involving a different coenzyme called FAD. So, I mean, the long names of these are not important. NAD stands for, well, the interesting side of NAD perhaps is that it's a nucleotide, and we've seen nucleotides before at AS, so it's the same old things that we see. NAD stands for nicotinamide adenine dinucleotide. Well, that's not important. The fact that it's a dinucleotide is interesting perhaps, but that's it, I think. The FAD stands for flavin adenine dinucleotide. The adenine you've heard before, and the idea of a nucleotide you've heard before, but that's it. You do not need to know the names. But because you see a difference, you see an N and an F, the temptation is to ask, well, what does it mean? I think we can park those. It's just different, okay? FAD is just a little bit different, and we do see a different effect when we compare what we get from NAD versus FAD, or what we get from reduced NAD versus reduced FAD, but we'll look at that later on. Finally, we got one last reaction. That 4-carbon compound is converted back to oxaloacetate, and here we go. We're back with NAD converted to reduced NAD, and that's it. So what are we getting here? I'll just go back a step and say, so look, by reforming oxaloacetate, it means that we can carry on this cycle. It can pick up another acetate fragment, form citric acid, and the same thing will happen again. So provided there's acetate available, we can continue to let this Krebs cycle continue. Right, so what do we got? So from glycolysis, we had 2 ATPs, 2 reduced NADs, and 2 pyruvates. Now from the Krebs, okay, so we're going to have 2 ATP molecules. Right, temptation is, where's that second ATP molecule coming from? There's only one. Yes, there is only one, but remember that this Krebs cycle and this link reaction happens once for every pyruvate molecule that's produced from glycolysis. And in glycolysis, there were 2 pyruvate molecules produced. So from the link and Krebs cycle, so the net gain from link and Krebs from one glucose molecule is 2 ATP molecules. We're doubling what we're seeing here because there are 2 pyruvates produced from each glucose molecule. We can do the same for reduced NADs. Count up the reduced NADs. 1, 2, 3, 4. Double it because it happens twice for each glucose molecule. And for FAD is 1, but we'll double it because it happens twice for each glucose molecule. So this is what we're left with. Right. Let's get those totals. So we've got 4 ATP molecules so far. We've got a total of 10 reduced NAD molecules. And we've got 2 reduced FAD molecules. Now the ATP molecules we're kind of done, aren't we? That's the goal of respiration, is to produce ATP. Brilliant. But we've still got these leftovers now. These from glycolysis, link and Krebs. We've got reduced NAD, reduced FAD. Those are the things that are going to go into the electron transport chain. Okay, that last stage. So let's have a look at the sort of thing that you might be asked here. So again, we're seeing something a bit bizarre because we're seeing a load of names that we're not familiar with. I'm going to give you another minute to have a little think what's going on here, trying to match up what you're seeing to what we've done in what we've just covered with the diagram that I showed you, and then we'll go through the questions. Okay, so a couple of things worth noting before we start to look at any questions. First thing, notice the way that NAD is represented. They refer to it as NAD plus converted into NADH plus H plus. Just refer to this as just reduced NAD. Okay, you can, that's what we would. So if we were to continue to refer to it like that, it's just cumbersome. So NADH plus H plus. We said that all the time. Reduced NAD just is much easier to say. And sometimes it's shown like it is here. Sometimes it's shown as reduced NAD. Sometimes it's shown as NADH. We might see examples of that in future slides now. So they're not showing the acetate being produced. They're just showing acetyl-CoA. They're not showing coenzyme A coming in. It's just acetyl-CoA. They got oxaloacetate to citrate, and now they've got these intermediates. They've got isocitrate, alpha-ketoglutarate, succinate, fumarate, malate, and that really serves to be quite confusing because you've got to think, well, hang on a minute, this is not the version I know. But you still have the same things appearing. You're still seeing carbon dioxide being released. You're still seeing, and being released twice, you're still seeing reduced NAD being produced, reduced NAD being produced, reduced FAD being produced. They're showing it as FADH2 there, and reduced NAD being produced, all in the same, if you like, in the same order, certainly. And we're seeing ATP being produced as well. So your job is to be able to track what's going on here. They ask you to begin with to determine how many carbons there are in the following Krebs cycle intermediates. You might be saying, well, I've got no idea how many in alpha ketoglutarate. I haven't studied that. But you don't need to, which is why we have to understand the reactions and track the reactions and see how the number of carbon changes, because we can use it ourselves. We know that isocitrate must have 6 carbons because there's no carbon being lost between citrate and isocitrate. Indeed, isocitrate is an isomer of citrate. And then we have a loss of carbons here. From isocitrate to alpha ketoglutarate, we're going to have a loss of carbon. So we know how many there are in citrate. It says use your own knowledge, and the diagram above. Your own knowledge should tell you that citrate has got 6 carbons. So 6 carbons for citrate. And then from isocitrate to alpha ketoglutarate, we've got a loss of CO2. Well, alpha ketoglutarate must have 5 carbons then. And from alpha ketoglutarate to succinate, sorry, we've got a loss of CO2. So succinate must have 4 carbons. So we don't need to know. We don't need to know how many it has. We can work it out. Explain precisely how you arrived at these figures. So this is taken from the mark scheme now on the right hand side. So knowing that citric acid is 4 carbon oxaloacetate plus 2 carbon acetyl or acetate. So that section there. That's where you get the first mark for saying how many there are in citrate. And then your second mark is just to sort of talk them through it really. There's a carbon lost, or CO2 is lost before the alpha ketoglutarate or from isocitrate. Or you could say isocitrate is decarboxylated. Okay, it's worth looking for these words on mark schemes. So you might say, well, yeah, I said that it was carbon dioxide lost from isocitrate, so I'm getting that mark. Yeah, fine. But we don't really care about how many marks you get right now, do we? Because what you're trying to do is learn. So it's all very well getting the marks, but really you're looking for opportunities to improve your understanding. So you can see that the fact that it says isocitrate is decarboxylated is a good opportunity for you to consider for yourself, okay, yeah, I could have said decarboxylated there. In order to get that state, there's 2 marks here. So the first mark is for saying the first point up there, oxaloacetate plus the acetyl. The second mark is for saying both of these things, that you're getting carbon dioxide lost and CO2 being lost from that, or CO2 being lost from alpha ketoglutarate or before succinate. So that section there, you see the CO2 lost there. Okay, let's have a look at another example here. So its diagram shows link reaction in the Krebs cycle. I'm going to give you another minute. It's a similar sort of thing it's asking, but let's have a go. So I'll give you a minute to see how you get on, and then I'll come back and give some feedback. Okay. So the first thing, quite nice, state precisely where in the cell the reactions take place, the Krebs cycle take place, as 2 marks. Good thing to spot, okay? And it says precisely, and it's in bold. Common mistake here would just be to say the matrix, or just to say the mitochondria, but actually you need both, mitochondrial matrix for both marks. It says on the diagram, use arrows marked CO2 to where carbon dioxide is removed. The sort of thing that pupils miss very often because there's no line underneath it. Just read the questions carefully. As it turns out, we're seeing something similar to the last question, isocitrate there. And then oxoglutarate, succinate, fumarate, malate. Okay, no big deal. Again, the intention here is to ensure that you understand. So we can see that from isocitrate to oxoglutarate, there's a loss of 1 carbon there. So I can draw CO2 on there. Same from oxoglutarate to succinate, there's a 5 to 4 carbon, I can draw CO2 on there. It says name the process by which carbon dioxide is removed. We should be familiar with this now, decarboxylation. And the role of the Krebs cycle and glycolysis is to generate reduced NAD to be used in ATP manufacture, something we'll look at next time. Describe the way in which reduced NAD is produced in the Krebs cycle. Well, there's 3 marks here. Not a particularly straightforward question, I don't think. So how is it produced? Well, as a result of dehydrogenation or dehydrogenase activity. I talked about the enzyme being a dehydrogenase as a result of dehydrogenation, or the removal, or which is the removal of hydrogen ions from the substrate from which it's taken. And then it's taken from a marked seam, so this is not the sort of thing I think I would have said, in a series of reactions or stepwise. 5 pairs of hydrogens, so it happens 5 times. It happens once in glycolysis, or you could say twice in glycolysis. Then you see it then elsewhere, obviously multiple times in the Krebs cycle. And then this, what is it referencing here? NAD to NADH2, or reduced NAD, or NADH plus H plus being formed. Again, they don't mind which one you use. That's why I prefer reduced NAD, because it avoids some of the more cumbersome ways of saying it. Okay, so again, we're looking at asking you to identify, you have to match up something new, a different way of showing the Krebs cycle with what you've seen before. Right, okay. We are at that stage now where we would now go on to the electron transport chain, but we are looking at the time. I think now is a great time to stop. So I will continue this presentation in 2 weeks' time, and we can go through the electron transport chain and look at the yield of ATP of each of the stages of respiration at the same time. Okay, so hope you found that useful. I hope you can come back now in 2 weeks and see the rest of that that presentation on respiration, and we can finish off the things we need to cover. Okay, bye-bye.